Thank you, Vivek, for the kind introduction. First of all, I want to thank the organizing committee for their kind invitation. Always a pleasure to come back to San Francisco. Also, great pleasure and honor to be part of this new wave and fast-moving research area that is viral ecology. It's really going fast, as you can see even here. If you compare the size of the room yesterday compared to the size of the room today, so it's going pretty well, right? So this is the outline of my talk. So I'm briefly going to talk to you about the phage collection. I'm glad to report that there are some people still doing phage biology. And then a little bit about how CRISPR-Cas works in bacteria and how now we're using it to edit phage genome. And I think this is the next wave of pretty cool research that's going to happen in the next few years. So I'm sure you all know that. Félix Derel was one of the co-discoverers of bacteriophages. I don't know if you know about this, but the word bacteriophages entered the literature in September 10th, 1917. So about 106 months ago, Félix Derel coined the term bacteriophages, and that's why we've been using it since that time. And in honor of Félix Derel, a professor at Université Laval in Quebec, Canada, Professor Ackerman, started to build a non-for-profit phage collection. He started this in 1982. Professor Ackerman retired in 2002, so 20 years later. Unfortunately, passed away last year. But we took over his collection in 2003, so 15 years ago. And the mission of the collection is still the same. Again, a non-for-profit organization is to conserve and distribute reference phages to foster research and education. So we have phages that are infecting over 135 bacterial species. And Denise Tremblay is the person handling all these different phages. So I would encourage you, if you want to do some phage biology, looking for a phage or reference phage, you can go on the website and see if we do have that. Just for your information, for the past 5 years, we have shipped phages to over 223 labs in 31 different countries. So again, phage biology is doing pretty well around the world. This is the number of requests we got the last few years. So it was going pretty well, and then we crashed here because we sort of lost our Canadian funding, and we had to increase the fees. So apparently, phage scientists didn't like that. So the number of requests went down, and now it's going up. Last year were the best year ever for the phage collection. So again, phage research is doing pretty well, and this is the request we got from the United States. So apparently, US researchers are, keeping the wave going. So that's great. Something I just want to touch base on, because we have been talking about yesterday and today about metagenomes and availability of data. And there's a little risk here going on in phage biology. We have been trying to increase the number of phage in our collection. As we have heard today, we have heard yesterday, there are new phages, new viruses that are being discovered, being isolated. And it'd be really important that those be made available to the scientific community. And if you ask that, scientists publish a new phage, and then you're requested on one-on-one basis, most of the time they will send it to you. But if we ask them to donate those phages to a collection, they're extremely reluctant. So there's a risk here. We have to be careful, and I would encourage people that if you find a new phage, to donate them to a collection, even in the United States, ATCC, or in Germany, or in France, or even us, if you want to send us to Canada. So again, there's a perceived risk here because it seems that we're not sharing as many phages as we should. So hopefully this will change in the next couple of years. All right, so CRISPR. I'm sure you all know by now that it is an adaptive microbial immune system. So bacteria, archaea are using this to defend against invaders like phage or plasmid. And I'm just briefly going to talk to you a little bit how this works inside of bacteria. You might have heard about the technology itself, but most of the time people don't know how it works inside of bacteria. So CRISPR, again, it's a locus that you're found mostly in genomes or bacterial genome. You'll have repeat, you have spacers. Those repeats are fairly short. In streptococci, which is the bacteria we'll talk to mostly today, it's only 36 base pairs. And there you have variable regions as well. And those variable regions are also very short. In streptococci, there are only 30 base pairs. And now we all know that they are matching phage or plasmid sequences. And these are really, these variable regions here, which are represented by color rectangles, are really strain specific. So you can track strains using those variable regions. And of course, those, if you find a locus, a CRISPR locus inside your bacterial genome, you're going to look next to it and you'll be looking for some genes. And if you find those genes, they'll be called CRISPR-associated genes, or they'll be called Cas genes. If you want more information on how you classify these genes, you can go talk to Eugen, because he developed this fantastic classification for Cas proteins. So now you can know if they belong to a certain class, a certain type, or certain subtype, depending on kind of Cas genes are found next, or most of the time next to a CRISPR array. And today, I will mostly talk about a type 2 or type 2A, because this is a system, it's a very simple system, and it's also the one that has probably most studied, because the Cas9 comes from a type 2A system. And if you want to know if your bacteria do carry a CRISPR or CRISPR-Cas system, you can go to a number of databases. I particularly like this one. In France, there's a bunch of genomes that have been annotated there. You can go and check if there's a CRISPR or Cas system that has been found in these genomes. And so far, most of the people agree that about half of the bacteria will carry at least one CRISPR-Cas system in their genome. And if you compare to restriction modification systems, about 95% of the bacteria will have a RM system. But in case of CRISPR, it's only about half of them in bacteria, a little bit close to 90 in the case of archaea. So how it works in bacteria, it's pretty simple. They're really, well, simple now. There are three main general steps, something called vaccination, adaptation, immunization step, where the phage will acquire DNA from the phage genome. And then after that, some RNA molecule will be produced, and that RNA molecule will be used to cleave the DNA or the RNA, depending on what type of system you're dealing with. So this is a very, if you want to show this in a lab, this is pretty simple, really. So this is what we call a plaque, right? A lawn of bacteria, and if you add a little bit of phage, you start having plaques. If you add a lot of phages, the lawn is starting to disappear. And if you add a lot of phages, then you go for a beer or two and come back 48 hours later, you get colonies that will start to emerge. And those colonies are called bacteriophage-sensitive mutants, as you probably all know by now. This is a fairly low frequency event, so most of the cells will actually die when they're infected by a virulent phages, but some cells will survive. And if you do this experiment in E. coli, for example, most of the time those BIM will actually be resistant to phage because they have a mutation at the cell surface receptor. But if you do this for streptococci, for example, Streptohermophilus, which is a food bacteria, most of those colonies that are resistant to phage are resistant because of CRISPR. And that's why we were able to find the function of CRISPR-Cas system in streptococci, because people working with E. coli, they could not get the CRISPR to actually acquire DNA and so forth, because receptors were most of the time mutated. And of course, then later we learned that the CRISPR system in E. coli is often repressed. But again, in streptococci, it was relatively easy. And now you can, if you get those colonies, you can run a PCR on a CRISPR array. And if you have an increase in the PCR size, it means you have acquired no new piece of DNA, most of the time at the 5' end of the CRISPR array. It's about 66 base pairs in streptococci, 30 from the repeat, and 36 from the repeat, and 30 from the spacers, and those are coming from the phage genome. So again, you've got the phage genome here, you got a phage, and those one colony has acquired one spacer, another colony has acquired a different spacer coming from a different area of the phage genome. And again, these two are resistant to phage. Now, so the mode of action, how this is working, we know pretty much. There's some still events that we don't understand, but most of the time, phage will infect, DNA will get in, and we know that Cas1 and Cas2 protein complex will actually pick up a piece of that phage DNA and again introduce it at the 5' end. And we now know by work done in Israel that this is actually this repeat that has been duplicated at the 5' end. Now you can do this with another phage and do this again. So you can do iteration of this, And if you do this again with another phage using one colony that had been resistant to the first phage, guess what? Another spacer will actually maybe appear, yes, again, at a 5' end. So it's like adding a page to a new book. You're adding a new spacer, again, most of the time at a 5' end. The most spacers you do have, the most phage resistant you will get, right? So the bacteria will be really resistant to phages. Is that, at least that's what's the dogma right now. The more spacer you have, the more chance you'll be resistant to bacteriophages. And again, you can follow this by PCR, your CRISPR array. Like I said, most of the time, the new spacer will be acquired at a 5' end, but I just want to highlight the fact that sometimes you can have spacer acquisition within the CRISPR array. So this is a publication we showed you, we published this last year. God, time is flying. A year ago, and when we showed that in Streptococcus thermophilus, sometimes spacers are actually acquired within the CRISPR array. But again, most of the time, it's at the 5' end. So the difference, so in bacteria, and that part sometimes is a bit difficult to understand, but for those that are not familiar with the CRISPR-Cas system, but in bacteria, you'll have the repeat, you've got the new spacer, the repeat, and the new spacer, and so forth. But if you look at the phage, the same sequence, now it's called a protospacer, meaning coming from, so it will be the exact same sequence from the spacer that have been acquired by the CRISPR array, but next to it, especially for type 1 and type 2 systems, not all of them, but some types, we'll have this little motif here called prospacer adjacent motif. And that motif is responsible for that piece of DNA being picked up by the CRISPR array. And again, this will be very important because later down the road, that piece of DNA will be cut by Cas9, but this won't be because there's no PAM in the repeat right here. Right? So there's a big difference. So it's a self, non-self recognition process inside the bacteria. This was done by Alain Deveau in my group. But I've been working with phage for a couple of decades now. And if you've been working with virulent phage like I did, we had an issue here because we were wondering how come a CRISPR array would actually pick up phage DNA and not lose the battle with the infection from the phage itself. So when we're dealing with Streptococcus phage we're working with are like the Usain Bolt of virology. They're really fast. They go inside the cell, take over the cell. 20 minutes, it's over. The cell has burst. New virions are released. So we were thinking, how can this happen, really? Is CRISPR that fast, really? Or Cas1 and Cas2 that fast? So for us, it was a timing issue here. So we're wondering if something else was going on. And Alex Hines, a postdoc in my lab, we tested this hypothesis that actually this was happening when the cell was infected by a defective phage, or a phage that was not replicating very efficiently. And I won't go into all the controls that Alex did, but we published a couple of years ago that when we did the same experiment I just showed you, when we were using defective phage as well, we were getting way more colonies that were resistant to bacteriophages. And it's really interesting because when you get your flu shot, right, to prevent influenza, we're getting flu shot from inactivated viruses. But apparently, bacteria might have been doing this for a long, long time using CRISPR-Cas system. So it's the same model, but we think it's happening mostly when cells are infected by defective phages. Something to keep in mind when you do metagenomic analysis, I guess. And what I've told you so far, we are very proud to say that for the past 5 years, our undergraduate students in microbiology are actually repeating this entire experiment I just showed you. So they're doing that in their senior years, and we have published a detailed protocol of if you want to implement that in your undergraduate classes. It's a fantastic tool, especially for seniors, because they need to understand virology, microbiology, genomics, bioinformatics, to really have a good grasp of how CRISPR-Cas system works in bacteria. And again, this is available for a lot of people, for everybody. So the next step is quite important. Obviously, it's the expression. Now you've got a new spacer in the array. What's going to happen? Very briefly, there's a promoter in front of the array here. So you're going to have a long RNA molecule. There's no transcripts. There's no gene in there. So, and then you've got, this will be processed depending on what type of system. So at the end of the day, you're going to get very short molecule, CRISPR RNA molecule between 40 to 38, 4 to 60 nucleotide. And these will be bound to Cas protein, and they will be inside the cell waiting for the next infection. All right? So what you do have in case of type 2, you'll have Cas9 that will be armed with a CRISPR RNA molecule. Part of it will be matching the spacer, only 21 out of 30, and part of it will be matching the repeat, and will be another transacting RNA molecule that will help Cas9 to bind all this inside the cell. So again, this is a surveillance complex waiting for another phage infection. So the next step, obviously, is what's happening when a new phage gets inside. It will, that we all know that will be cleaved by the Cas proteins, and that you can see by two ways. One way is doing a classical plaque assays. So you get colonies that have acquired new spacers, you grow them, you test them for phage resistance. This is the wild type phage sensitive. This one has acquired one spacer, resistant to one phage. This one has acquired two spacers, resistant to two phages, as you can see very nicely on this plate. And you can go inside the cell and then look at what's happening of the fate of DNA, for example, and we know it's not cleaved. And the type 2 will actually cleave the DNA, a 3 nucleotide next to the PAM motif I just showed you earlier. So this is how the phage will actually be, the bacteria will actually be resistant to phage by cutting its DNA. And that will be obviously very important for the phage genome editing part I will talk to you about at the end of the day. So what you do have, now you got a CRISPR array that's producing a very short RNA molecule that will be bind to Cas9 in the case of type 2. And when a new phage DNA gets in, there will be a base pairing here between the part of the RNA molecule that will bind to the one strand of the DNA, and then Cas9 will actually cut it with its two main domain, and then that's the end of the phage genome, or the phage replication process. So at that time, the bacteria wins, the phage is actually inactivated. But, as Simon was talking this morning, phage are the most abundant biological entity on the planet. We have heard this a few times already yesterday. And I mean, if you're the most abundant, you do need to have a few tricks to be on top of this, right? And of course, phage are able to bypass the CRISPR-Cas system at least two ways. They can mutate. So this is an example here where we picked 3 different plaques, very rare events again. Those plaques were actually resistant to the spacer that had been acquired, and sure enough, the mutation was right within the protospacer or within the protospacer adjacent motif. So again, you need only one single nucleotide. In these particular phages, they have 35,000 base pair. Only one nucleotide is enough to bypass the CRISPR-Cas system. Of course, if you have 2 spacers, you will need 2 mutation. It's much more difficult at that time. And Bruno Martel, a master student in my group, analyzed 382 phage mutants here. And essentially, you need a mutation, but really at the 3' end or within the PAM motif. If you have mutation at the middle of the 5' end, it doesn't provide resistance or way of bypassing the CRISPR-Cas system. And we have heard already this morning about anti-CRISPR, and this work was done again in Canada at the University of Toronto. Joe Denomi. Joe now is at UCSF here in San Francisco. But Joe, when he was a grad student, he found anti-CRISPR proteins, so phage-producing proteins that will block Cas proteins, blocking the cutting and everything. So this was found in a prophage of Pseudomonas. Recently, we actually found an anti-CRISPR protein in our streptococcal phages, in virulent phages. We have analyzed 250 different genomes, and this anti-CRISPR protein is found in about 5 to 6%. So it's not very common in those virulent phages. We just discovered another anti-CRISPR protein that is much more common in Streptomophilus phages. So it seems that we're starting to selecting phages that are able to bypass the CRISPR-Cas system in the environment. And these are really easy to pick up, at least for us. So this is a phage that I just showed you. You can get colonies that are resistant to phage, but these anti-CRISPR carrying phages, you can get BIMs. So it seems that it's not acquiring any spacer, and those colonies are not CRISPR BIMs, there's something else. There's mutations elsewhere on the bacterial genomes. As we have heard yesterday, this can be happening quite often. So I'm going to quickly pass those. And of course, we have cloned this and so forth, and try to understand how this works into the phage biology and in the CRISPR. So how can we use all this information to study phage biology here? So as you all probably know, if you look at the phage genome, about half of its genome is fairly well known in the sense that you'll know it's a structural, genes coding for structural proteins. Many proteins, we have solved the structures, we have a very good idea of all these proteins are working. But if you're looking at the other half of the phage genome, really, that part is really a black box. There's really many genes there that we do not know what they're doing. Their functions, sometimes you'll see some recombinases genes and so forth, but most of the time you do not know. And those, when the phage infects a cell, those genes are the first one that will get inside the cell. And most of the time, these are genes that will redirect the bacterial metabolism for phage purposes, but I'm pretty sure there are proteins also that are blocking defense mechanisms or something else. And of course, anti-CRISPR, the ones that we have found, are exactly found in this area that is unknown. So for us, there's an interest of understanding this region. And if you're working with virulent phages, there's not really many ways to mutate them. And that enter the CRISPR-Cas system. And you can do this two ways. It will depend if your strain carries a CRISPR-Cas system inside its genome. I've told you about half of them do have a CRISPR, the other half doesn't. So if it does, you can use the native system to your advantage to mutate a virulent phage genome. And if you doesn't have one, you then try to introduce the famous Streptococcus pyogenes Cas9 into bacteria, and this is what Marie-Laurence Lemay has done in my group, and Bruno also has done it using the native system of Streptococci. So to do this, there's two requirements, really. You need an interference system, and you need an editing template because we're going to use homologous recombination to generate those mutated phage. So again, the phage will get in. You need a way to cut the phage genome. So you need to ask the question, is there an active CRISPR-Cas system inside my strain? If the answer is yes, then you only need to provide a plasmid with a repeat spacer, the spacer targeting a specific region of the phage genome, and another repeat. And this will provide resistance to your phage, and that will be the first part of the system. All right? So using the native endogenous Cas protein inside your bacteria. If it doesn't, again, carry one, you can introduce the entire system, and that's what Marie-Laurence did. We did it in Lactococcus, a different bacteria that does not carry a CRISPR-Cas system. And then you will introduce, again, this repeat, the spacer and the repeat, in the Cas9 to, again, provide phage resistance. So to design your spacer, you're going to look at your genome, you're going to look at the gene of interest, you're going to zero in, and then you'll look if there's a PAM motif next to it. If it does, then you'll take that piece of protospacer DNA, clone it inside your plasmid, and then you generate a plasmid with the spacer. And that construction, again, will provide phage resistance into your bacteria, as I showed you earlier. The second combination is an editing plasmid. In this time, it will be the same way, no matter if your bacteria carries a CRISPR-Cas system inside its cell, it's the same way of doing it. And how we do it using homologous recombination. So this is, like again, the phage genome. This is the area you would like to mutate. Either you would like to inactivate, delete that gene. Either you want to introduce a point mutation. Either you want to tag it with a histag to pull down assays to understand phage protein-protein interaction and so forth. What really important is you do need to clone regions that are flanking the area you want to remove. So you're going to generate by PCR and some trick in the lab, Gibson assembly, you're going to generate a plasmid with homologous recombination that will use homologous recombination to replace that middle part here inside the phage genome. How this will work? Well, the phage will get inside that strain that will carry your interference plasmid, your editing plasmid. So the phage will get in. It will be cut by the CRISPR-Cas system, again, endogenous or heterologous. The phage, at this point, will die or will be non-infectious. So it needs to repair itself. It can do it if you provide a recombination template. You'll have a homologous recombination. You will introduce whatever you want to introduce, either a deleted gene, a tag, or a point mutation, and then you have yourself an engineered phage. And that engineered phage is, if you design this correctly, it will no longer be cut by the CRISPR-Cas system, and then you get yourself a phage mutant. So this is a two case here, Strep thermophilus, which has a native system. This work was done by Ariane Renaud in my group. So she has inactivated 4 different, in this gel, she has deleted 4 non-essential phage genes. So now you can check what's happening at the phage biology level, what's the impact on the replication of the phage, what's the impact with the host proteins, and so forth. There's many more that have been inactivated at this time, but this gel is only presenting 4. It's working beautifully using the native system. And this, if you introduce an heterologous system, Streptococcus, Lactococcus lactis, we introduced the Streptococcus pyogenes Cas9 system, and this work was done by Marie-Laurence, as we cannot see here. She inactivated 4 different genes, and she was even able to generate a mutant in those 4 genes were actually being inactivated. She generated a phage with 8% of its genes that have been inactivated, and the phage was still fine, able to replicate. And obviously, we're studying what's happening at the phage-host interaction level. Interestingly, using this and the other system I just showed you, there was no off-target issue whatsoever, so the phage were mutated at the right position, and that was it. And you're able to study this. And these new tools are kind of cool because it allows us for the first time to study something different when you're using virulent phage. Again, this is very difficult to modify. So this is just an example with one phage that we have inactivated one gene, one phage, one gene. So again, I'm go back to the same slide. We got really very interesting results. So this is what we would take phage P2, for example, Lactococcus, typical assay, just the same slide I showed you. You get BIM, few BIMs. And if you do the same experiment with phage P2 delta 47, the gene that has been inactivated, you get this. You get loads of colonies. So it seems that that gene somehow in the phage is preventing the bacteria to evolve to becoming resistant. So it's really interesting to us, just one gene. Obviously, so again, the presence of OR47 is reducing the development of BIM, which makes sense from a phage perspective because there's this protein that's preventing the bacteria to evolve very rapidly. We have solved the structure of this protein. We think it is a regulator. We're currently studying, obviously, all these little colonies here and comparing to these other. But it's a fairly fantastic tool, and you can imagine whatever you want. You can introduce genes, you can delete genes, you can tag them, and it's really, really neat, at least from my perspective. So I'm running out of time. Conclusion here. Well, I think I've showed you how it works in bacteria. You can vaccinate your strain. You can produce RNA molecule that will be bind to Cas protein, and then it can cut phage DNA. How the strains are, I keep talking about Streptococcus thermophilus. This is a bacteria that is used to make cheese, to make yogurt. So if you're eating cheese and if you're eating yogurt, you're eating Cas9 proteins. It's not pyogenes, it's Strep thermophilus. So most of the strains that are used by the dairy industry today are CRISPRized. Strep thermophilus has a CRISPR-Cas system naturally. Phage are the enemy in the milk fermentation industry because if you are attacking your cultures, your good bacteria, you won't have good cheese, you won't have yogurt. So it's very important to have bacterial strains that are resistant to phage. This is a natural system I just show you. So you can, most of the strains that are out there are resistant to some of the phage, at least to the phage that are known through CRISPR. And that's why we're selecting now in the industry phage that are carrying these anti-CRISPR and all these other means of bypassing the CRISPR system. As we have heard already, the spacers in the CRISPR array can be used to track your strain. They can track phage pass infection. You can probably predict phage host, habitat, as David mentioned as well. So if you're looking at a strain, looking at the CRISPR array, looking at the spacers, you can have a very good idea of the habitat, where it's coming from. Very useful in epidemiological studies if you want to know where that strain came from and so forth. Especially in the food business, food safety business, if you want to track a strain that has caused infection in some vegetables in Wisconsin, and you want to know if in Montreal or in Quebec City we have the same strain, same problem, you can look at the spacers content of the strain, and if it's the same spacer content, pretty good chance you have the same strain. So it's very useful, again, in this area. And really, I believe that we are entering this fantastic area as well. Now we have this fantastic tool, I think, to modify pretty much everything we want. We can generate phages, we can mutate them, we can study how they will interact with the phage, with the bacteria, and I think we're going to have this phage craze in a very short time if it's not started already. And I certainly I'm here to talk to you about all this, but of course we all have the best group, right? But I'm right. So I really have a fantastic group, and I want to acknowledge especially Denise here, which is sitting there, which is in charge again of the phage collection, and Marie-Laurence also, which work on the genome editing tool, and all the other people, and of course all the funding agency in Canada and CIRC-CIHR and Canada Research Chair and all the different industries. So on that, I want to thank you for your for being here, and I'll be happy to answer any of your questions. Thank you very much.